Adam, that's a lovely introduction. Okay, I'll try and share my screen. Just let me know if you can see it. Oh, hang on. Which one's the right one to share? Okay, let me try this. Can you see that? Yeah. Okay.
Yes.
Perfect. Just yeah. Okay. Okay. All good. Thank you very much indeed for that very kind and flattering introduction, Adam. And thanks very much for the invitation. It's great to be able to speak to you about my work. I've got a bit of a cold, so apologies if I sniffle or cough at any point, and do unmute and yell at me if you can't hear what I'm saying. I'm going to talk to you today about really kind of my experiences working in the kind of field of telomere dynamics, and try to offer some insight into the question that's been bothering me quite a lot lately, which is telomere length a useful biomarker in evolution and ecology? And to start with, I just want to give a brief introduction to kind of my own research interests, which Adam's actually done a very good job of doing already. But I'm an evolutionary ecologist, and I suppose I'm principally interested in understanding how genes in the environment shape variation in phenotypes and investment in different life history traits, and how they in turn give rise to variation in demographic rates and fitness, and how they drive important ecological and evolutionary processes. And I work with long-term individual-based studies, data sets collected from those studies in wild vertebrates. Principally, I've worked on these long-term studies of wild ungulates. And one process that I've been particularly interested in trying to understand in these kind of natural systems is senescence. So the physiological deterioration of function in later life that we see so clearly in our own species and many animals in captivity and in the lab. And this plots from a review that myself and collaborators published a few years ago now, in which we tried to survey the ecological literature and collate studies that provided some evidence for patterns consistent with senescence in the wild. And what you can see is that the kind of amount of evidence has been accumulating quite rapidly. By about 2012, we were able to find evidence for manifestations of senescence in around 180 species. So this is really suggesting there is quite clear evidence that you can see, observe, and study demographic senescence in the wild. And I think a lot of the reason for this rapid accumulation of data and interest is down to the emergence and development of long-term individual-based studies in the field. You can see in this plot that most of the evidence is coming from birds and mammals for obvious reasons. But I think over the last few years, we are seeing a growing number of studies, of these kind of longitudinal studies in the field of both other kinds of vertebrates and invertebrates that provide evidence and insight into the process of senescence in the wild. One very important thing that I think is very clear from this rapidly emerging literature is that we observe very considerable variation in demographic aging patterns and senescence patterns, both among species and among individuals within populations. And it's this variation that I've been particularly motivated to try and understand, particularly the variation we see among individuals within wild populations. And this is what demographic senescence looks like in two of the study systems that I've been most closely involved with over the course of my career. In the top right, you have age-specific survival probabilities in males and females from a long-term study of the red deer on the Isle of Rum. And the bottom left, the long-term study of sowy sheep on St Kilda, which Adam mentioned already, I've been closely involved with over the last little while. What you can see in both plots is that from a certain point in adulthood, in the deer it's around 9 years of age, in the sheep maybe 5 or 6, the probability of survival over the subsequent year, over the following winter, starts to decline. And you can see in the deer there's quite a dramatic difference between the sexes. The males in blue there show a much sharper decline in senescence than females from that point. It's not so clear in the sheep, but you can see very clearly that the probability of survival is declining. There's a very clear manifestation of actuarial senescence here. And what's really, I think, important and interesting about these systems, they're unmanaged and unpredated systems that are otherwise subject to the pressures of natural selection. And a really quite high proportion of individuals that survive their first year in these two populations will go on to survive into these senescent age brackets where we see demographic senescence. So it really gives us a good opportunity to study and try to understand variation in senescence in the natural environment. My interest in senescence has really kind of started by a kind of postdoc that I took up in Cambridge when I finished my PhD. And this was working with Tim Clutton-Brock in Cambridge, but also with Luska Crook in Edinburgh, and Alistair Wilson, who at that time was in Edinburgh and subsequently moved to the University of Exeter. And this really used the astonishingly detailed longitudinal information that had been collected over what's now nearly 50 years in this remarkable longitudinal study. So in the study area of deer on the Isle of Rum, newborn calves are caught usually within a week of birth. They're marked so they can be individually recognized, and they're very, very closely monitored throughout the year by very skilled, very dedicated field researchers who historically have always lived in the study area. That field work was initiated and led for many years by Fiona Guinness, but during the period that I've been involved in the project, it's principally been done by a husband and wife fieldwork team, Ali and Sean Morris. And the kind of contribution of these guys and the kind of skill and dedication that's involved in kind of generating and maintaining these amazing long-term data sets is really, I think it's difficult to understate and obviously acknowledge all of these people, but very mindful of the amazing work that goes on in the field here. What we did was really to try and use the very high quality data on age-specific mortality and reproductive performance to try to ask some questions which hadn't been at that time, this is about sort of 2006, 2007, very well explored in the context of wild animals. And we were able to show that senescence rates, variation in them had a genetic basis in the wild, and also that early life environmental conditions could impact the rate of aging much later on in adulthood. We're also able to provide support for predictions from evolutionary theories of aging that males in these kinds of systems should senesce faster than females, and also that increased reproductive investment in early life should predict more rapid rates of senescence in later life. I think following on from that postdoc in Cambridge, something that's always really interested me and become an increasing focus is trying to understand what underpins demographic senescence in the wild. Senescence is by definition a physiological process. It's a very complex and multifaceted process of deterioration that varies hugely in all kinds of different ways. So different physiological processes change and behaviors change, life history changes in different ways, and often different traits or measures will senesce in different ways within a given species or population. So really, I've been very motivated to try to understand what kind of sits in between the effects of genes and environment and demographic senescence patterns in the wild. This is another plot taken from the 2013 review we did. What this really illustrates is that at that point, around 8 years ago, from surveying the ecological literature, most studies that we could find in wild animals had focused understandably on demographic variation, so survival, annual survival, or reproductive performance measures. There was very little or relatively little attention on what might be going on in late life in other kinds of traits. But actually since then, I think there's been a kind of real surge of interest and some very interesting studies emerging that kind of delve underneath demographic rates and reproductive performance. So there've been some very interesting studies on different birds and mammals which look at changes in behavior in late life, and particularly changes in foraging behavior. I think there's actually a really rapidly growing and establishing body of literature looking at immune function in late life in all kinds of different vertebrates in the wild. And I think there is quite coherent evidence now that in many wild systems, we see age-related declines in immune function, and these might have a very important role to play in demographic senescence in the wild. Now, another biomarker that has been the focus of a great deal of interest in evolution and ecology lately is telomere length. And it's this just kind of physiological or molecular marker that I want to focus on for the rest of the talk. And what I really want to try and do is first very briefly and very crudely tell you what telomeres are and what they do at kind of cellular level, and explain why they've become a biomarker of such interest in the context of kind of human research, particularly biomedical and epidemiological studies. I want to give a kind of personal perspective on the kind of emerging interest and the challenges of kind of applying and studying this biomarker telomere length within evolutionary ecology, and try and use what I think are some really fantastic meta-analysis using the ecological data that's emerged recently to try and paint a picture of where we're at in terms of our understanding of telomere length. And then I want to talk through some of our most recent work looking at telomere dynamics in the Sow sheep on St Kilda, where we've addressed the repeatability at the individual level and the heritability of telomere length, and also tried to delve into and understand the processes responsible for quite a widely observed association between telomere length and subsequent survival. So very crudely, what are telomeres? They are repetitive sequences, usually of this TTA-GGG motif that sit at the end of the chromosomes of most eukaryote species. They have a very important role in protecting linear chromosomes. They are thought to have a very important role in maintaining genomic integrity and stability. Telomeres are eroded, so telomere length is reduced by cell division, so a small amount of telomeres lost at every cell division due to the so-called end replication problem. And it's also known that various forms of cellular stress, particularly oxidative stress, particularly target telomeres and can reduce telomere length. And there are processes that can restore telomeres, particularly the enzyme telomerase, which can lengthen telomere length. And I think very importantly, having critically short telomeres is known to trigger cellular senescence. So you can see that these telomeres have a very important role in genomic integrity, and also have a role in controlling cellular senescence. So telomere length and telomere attrition, I think, are quite widely accepted as a very important part of the suite of cellular and molecular processes that biogerontologists now think are responsible for aging in quite a general way. So this is from a paper from a few years ago, which identified 9 so-called hallmarks of aging. And you can see there in the kind of red chunk that telomere attrition is one of those 9 hallmarks. A lot of effort and interest has also gone on, in particular in humans, in trying to measure sort of some average telomere length at the kind of organismal level, and study how variation in that average telomere length or telomere attrition over time might relate to things that we care about in terms of disease and mortality outcomes in later life. And there is, as I'll show you in a bit, a sort of mounting evidence and kind of call for the idea that telomere attrition at that kind of level might be associated with the risk of late onset disease and follow-up mortality in humans. And there are various kind of ideas of what telomere length and telomere attrition might be telling us in the literature. It's certainly talked quite often about as a kind of a marker of biological aging, so some sort of indicator of the biological state in a broad sense of an individual in relation to this kind of broad physiological decline that we think of as aging or senescence. You've also seen it kind of referred to as a kind of marker of the so-called exposome. So this catch-all marker of the pressures and the stresses that individuals have experienced over their lifetime from the environment. From my point of view, I don't think there's very much support for a causal relationship between telomere length and aging at the whole organism level at the moment. So I think a lot of what we're talking about are kind of correlated relationships between telomere length and other important processes, but that doesn't mean that telomere length and telomere attrition couldn't provide important insights and be an important biomarker of things that we care about. And the human literature, I think, is kind of really suggesting some quite important and interesting things about how average measures of telomere length in an individual behave and what it can tell us. One very important thing to bear in mind when we talk about average telomere length measured from a sample taken from an individual is that usually, for obvious reasons, these need to be taken in a kind of non-destructive way, and most of the studies will use blood. There is a kind of move towards using saliva and kind of buccal samples as well in humans, but I think most of the studies I'm going to refer to here look at blood telomere length. Now, because we as mammals don't have nucleated red blood cells, that means that the DNA you get from blood is coming from the leukocytes, this diverse group of white blood cells that are involved in our immune response. So when we're talking about telomere length or leukocyte telomere length, that's really what we mean. It's the average telomere length across these cells. What I think is emerging, or is kind of generally seems to be the consensus in the human literature, is that telomere length measured in this way shows what's called a biphasic decline with age. So there's a kind of fairly rapid decline over the first few decades of life in telomere length, and then a slower but still significant decline thereafter into much older ages. I think there's quite a kind of coherent sense in the literature that various different forms of behavior and experience that might be deemed stressful are associated with shorter telomere length. So this is a recent meta-analysis that shows this. So records or evidence of prior sort of stressful events tend to predict short telomere length. There's also evidence from a number of human studies that telomere length is kind of repeatable over the course of an individual's lifetime, and also that it may be highly heritable. Heritability estimates in humans vary a little bit, but they often found to be quite high. In this particular meta-analysis, it was found to be very high, at around 70%. So there's a genetic contribution to variation in telomere length. And perhaps most importantly, a number of studies in this meta-analysis kind of really illustrates that, show that having short telomere length at kind of point of measurement is, generally speaking, on average, associated with an increased subsequent mortality risk. You can see a lot of variation in the estimates from the different studies in that meta-analysis, but overall, there is that association between shorter telomere length and increased mortality risk. So lots of interesting associations that we're seeing across the human literature. I think in the kind of early to mid 2000s, and a relatively small number of groups working on kind of ecological and evolutionary questions started to get interested in telomere length, particularly Pat Monahan's group in Glasgow and Mark Hausman's group in the US. And they, between them, produced this, I think, really important and exciting sort of perspective on telomere length, kind of putting it forward as a potentially really interesting biomarker that could tell us about the relationship between lifestyle and life experience and lifespan and aging. And they kind of framed this very much in terms of a trade-off on the one hand between having long telomere length and therefore having raised kind of cell proliferative potential and perhaps delayed or reduced senescence, but at the kind of cost of the increased risk of sort of runaway cell proliferation and cancer. And on the other hand, eroded shorter telomere lengths, limiting cell replicative potential and therefore lifespan. So I think this was framed at the time as being quite kind of causal, and maybe we've come away from that a little bit, but I think it's a very, very influential and important paper, which really kind of set the scene of a lot that followed. I can remember, I don't know whether Britt remembers this as well. I can remember as I was becoming, I started talking to Pat and started becoming interested in studying telomere length with some of the systems I work on. This paper came out, led by Torsten Horn in 2010. And this was I think a very important and very interesting review of the work that had gone on in ecology and evolution. There weren't that many studies out there, but there was some work going on, particularly in birds. And it was very highly critical of some of the work that had gone on, and particularly critical of the way people were approaching some of the methodological issues. And as you can see this quote, they're really highlighting that a lot of the issues in this kind of early literature on telomere length and ecology and evolution are mirroring some issues that have been a problem within the human telomere literature for quite a while. I think it's important to sort of just reflect on some of those issues before I talk more generally about what's really become a very big and very exciting literature on telomere length in ecology and evolution. And a lot of it really boils down to an issue about methodology and methodological differences. Fundamentally, at the heart of this is a kind of dichotomy between two very widely applied approaches to measuring telomere length in the literature. And this is true in both the human literature and in the broader ecological literature. The so-called telomere restriction fragment, or TRF analysis, and the qPCR method. It's beyond the scope and probably beyond my capabilities to explain the differences between these methods in any kind of detail. I'm happy to try and talk about them with you afterwards if you want. But essentially, the TRF, which is kind of southern blot-based method, is, well, certainly from experiences we've had in my lab, a technically very challenging method to set up and get running in kind of robust and repeatable way. But it's broadly thought of as a kind of gold standard method. It tends to produce very high technical repeatabilities, and it's one of the kind of original methods. So it's thought of as being kind of very robust and repeatable. It produces this kind of smears of telomeric DNA, which you can relate to a DNA ladder for any given sample, and that allows you to sort of generate data that is in kind of clear and comparable kilobase units in terms of telomere length. So it is kind of real absolute units of telomere length. On the other hand, the qPCR approach, which is now, I think, the dominant approach in most literatures interested in telomere length, it's faster and potentially much higher throughput than TRF. In terms of technical repeatability, it's often a lot noisier. The studies out there that have compared the two methods directly tend to find that it has much lower technical repeatabilities. And because of the way it works, you're essentially expressing amplification of telomeric DNA against the amplification of a non-variable copy number control gene. What you're getting is telomere length, or relative telomere length, expressed in arbitrary units that are very specific to the particular study and conditions of measurements. They're not directly comparable across studies, and they don't tell you anything about kind of actual telomere length in kilobase units. There are other methods, very powerful methods. I think things like Flowfish and Stellar, they're even more involved than TRF, and they tend not to be very widely used in the ecological literature, so I'm not going to discuss them further. But what I am going to sort of say is that even with this kind of quite tricky dichotomy in approaches, there are further, I think, quite loud and angry elephants in the room here. In fact, you could probably argue that the telomere room is pretty packed with elephants. One issue is that I've been talking about telomeres as kind of these repetitive sort of runs of DNA that are at the ends of chromosomes, but in fact, many species have sometimes quite considerable amounts of telomeric sequence within the chromosomes, so-called interstitial telomeres. And this can be a big problem. So the kind of classical southern blot-based denaturing TRF and qPCR do not differentiate. So they will measure both end and interstitial telomeres. And obviously, we are interested predominantly in the end telomeres. There's a kind of source of quite concerning bias there. This plot shows some microscopy done on chromosomes from different reptile species that shows actually that species vary quite a lot in the amount of interstitial telomeres that they have. Important to acknowledge that the so-called in-gel TRF method, which I think was developed by Mark Hausman in the US and is used in a small number of labs at the moment within ecology, principally Mark's and Simon Verhulst's lab. I think also Pat now has this working in her lab. Britt might tell me that she's got it working in her lab as well. We were never able to get this working in our own lab, but the key thing about this approach is that it actually is able to target just the end telomeres. So this approach does not measure interstitial telomeres, but it is also very technically challenging to run. As if that wasn't enough, there's another issue in the kind of ecological literature, particularly when it comes to studies of vertebrates, which most of the studies that we talk about are. And it's a question of whether we're actually measuring DNA and telomeres in red or white blood cells. So as I mentioned before, mammals don't have nucleated red blood cells. So when you take a blood sample and extract DNA, you're getting DNA and you're measuring telomere length in the white blood cell fraction. In other vertebrate groups, the red blood cells are nucleated, and because they outnumber white blood cells by such a tremendous degree, the signal you get, the DNA you get, and the telomere length you measure are going to be basically red blood cells. So we are measuring telomere length in different kinds of cells, and actually at the moment we really don't know very much about the differences in telomere lengths between these different cell types and how they could be influencing results that we get across different kinds of systems. So around this kind of point where that paper came out in 2010, I was just starting to enter into discussions and think about measuring telomere length in our long-term study of sow sheep with Josephine Pemberton, who's a colleague in Edinburgh who's been kind of managing and running this project for many years now. And we'd started to form a collaboration with two really wonderful cell biologists and telomere biologists, Lee Harrington, who at that time was in the Institute of Cell Biology in Edinburgh and subsequently moved to Montreal, and Jen Fairley, who was running Lee's lab in Edinburgh. And Jen was starting to pilot using samples we collected from the sheep, both qPCR and TRF methods. And around this point, I've met Pat Monahan at various kind of meetings and workshops and started to talk about the kind of issues with methodology and some of the problems that were coming up in the literature and some of the debates that were emerging. And Pat hit upon the idea of trying to bring together the groups that at the time were involved in working on telomere length. Britt, you're in this photo at the time, and also bring in some experts from the field of human epidemiology and cell biology, and try and sort of talk through some of these issues as methodological issues that were really bothering people at the time. And we managed to get funding for a small workshop that was held in the village of Drimmen near the banks of Loch Lomond, which definitely was one of the most sort of interesting and rewarding scientific meetings I've been to. And there was a real sense of, although we were worried there would be quite a lot of debate and argument in this meeting, there was actually a real sense of everyone wanting to find consensus and really evaluate the pros and cons of different approaches and try and identify best practice. And off the back of that, Pat was able to get funding from the Leverhulme Trust to set up a telomere network, for which we ran meetings every year, initially in Drimmen, and then when the network became too big, we moved it to a larger hotel in Edinburgh, and that was running annually until the pandemic took the wheels off pretty much everything. But again, I think what's been really good about this from my point of view is it's kind of really bringing together the ecological community working and interested in telomere length with experts in cell biology and kind of other fields. I think it's really been important in keeping the field together and helping people understand the pros and cons of different technical issues. We're emerging from this, from the Drimmen workshop, we ended up writing a kind of sort of method synthesis piece, which I think helped try to provide some sort of balance and consensus view on the problems and the kind of pros and cons of the different methodologies for evolutionary biologists and ecologists, and more recently kind of co-edited an issue of Phil Tran's The Royal Society B, which I think really just showcases some of the amazing different work that's going on, different kinds of systems and settings looking at telomere dynamics. And I think what's emerging from this kind of very, very rapidly growing field of studies of telomere dynamics in kind of ecological and evolutionary contexts can be seen in some really nice meta-analysis that people are starting to do from the rapidly emerging data and kind of published studies. So this is, I think, a really nice meta-analysis that was published a few years ago from Marta Zulkin's lab in Poland, and it's similar to something I've shown previously looking at human studies, suggesting that different kinds of environmental variables that you might putatively sort of think of as sort of stresses, although you could argue about whether they really fall under the umbrella term of stress, are associated with shortened telomere length. And you can see there's a kind of clear overall effect, and generally across different kinds of environmental stresses or forces, that there's a kind of relatively coherent and clear effect. So that's consistent with what people see in humans and suggests that telomere length might be reflecting something about the environment. This is a manuscript that's been published on a kind of online preprint, and I'm presuming it's going to come out be published somewhere very soon, which looks at across studies of, this is non-mammalian vertebrate studies, at the repeatability of telomere length. So these are from longitudinal studies, and it really addresses how repeatable telomere length is across repeated measures at the individual level. So that the kind of top panel there is TRF studies and the bottom is qPCR. You can see there's far more work being done using qPCR, and you can also see very clearly a very pronounced methods difference. There's a lot of variation in repeatability. The averages are quite high, suggesting there is overall suggesting that telomere length is quite repeatable, but there's a very marked difference between methods. So TRF studies are providing much higher estimates of repeatability. It looks like over 0.75, which is massive. There's less data on the heritability because this would require quite detailed information on the population pedigree and genetic relatedness structure to try and estimate the genetic contribution to repeatability. But this very recent paper by Christina Bausch from Simon Verhulst's lab does a really good job of reviewing it. I think it's suggesting the same kind of thing is going on, that generally there's quite a lot of variation. Overall, there does seem to be evidence that telomere length is heritable, and there's a methods effect. It looks like TRF studies are providing higher estimates of heritability than qPCR studies. This might just be to do with the higher technical repeatability, but it's kind of highlighting the issue with methods that we have here in this literature. Kind of critical property of telomere length is its potential predictive power of things that we care about from an ecological and evolutionary point of view, particularly survival. And this is from a meta-analysis I was involved with. It was led by Rachel Wilburn, and Jelle Bunnekamp, who's now in Glasgow, was heavily involved in this, where we were able to get raw data from the studies that we could find in the literature that looked at this association. Overall, across these studies, we did find a weak association in the expected direction between telomere length and mortality risk. So individuals with shorter telomeres had increased mortality risk, lower survival probability. But you can see that there is a really quite dramatic spread of estimates across these different studies in this forest plot. You can also see, if you look at the number of studies there that aren't circles, that's birds, that there's very limited data outside of birds at this point anyway. There's a really difficult and surprising methods effect where we found evidence that qPCR studies are actually providing a stronger signal than TRF studies, and we really don't know why that should be. And there's also quite worrying evidence in this meta-analysis for publication bias, a suggestion that people are not publishing sort of analyses where they don't find an association between telomere length and mortality. So overall, it's kind of backing this suggestion that telomere length might predict survival and mortality risk, but there's quite a lot of things here that we don't understand. And again, this kind of worrying manifestation of a methods effect. This relationship between telomere length and survival is something we've been very interested in trying to probe in more detail in our long-term study of sow sheep. We have previously shown it with smaller longitudinal data sets that we can find that relationship, that short telomeres do predict reduced follow-up survival. But the literature suggests that actually there's two different kinds of processes or kind of conceptual models that might explain that relationship. I'm not invoking telomere length or telomere shortening as a causal driver of reduced survival probability, just trying to understand why through correlated effects it might predict survival. The first of these two conceptual models of what's driving the relationship, as I'm going to try and present them to you, I don't know if you'll find this convincing, is what I'm calling the exposome model. So here we might imagine that recent or cumulative detrimental effects of the environment are being reflected in telomere attrition. So telomeres get shorter, and that's correlated, it's correlated with increased mortality risk. So basically here, telomere attrition is reflecting kind of harsh, challenging environmental conditions. That's why it's predicting mortality. And this suggests that the relationship between telomere length and survival should exist at the kind of within individual level. So it should be associated with reductions, loss of telomere length over relatively short periods of time within the lifespan of an individual. And there is some evidence, say, from birds for this kind of relationship, that telomere attrition over a certain period of time is associated with mortality. Most of these kinds of studies, however, focus only on quite early life, so the nestling period and telomere attrition, that period. They're not looking across the entire lifespan. The other sort of conceptual model relating to the relationship between telomere length and survival or longevity is what I'm calling the kind of frailty or robustness model. This is also consistent with a sort of selective disappearance model. Essentially something about an individual's genetic makeup or its early life environmental conditions, or something consistent about its environmental conditions, was driving consistent differences in average telomere length. So some individuals have longer telomeres consistently on average across their lifetime, and others have shorter. That would be what we would expect if telomere length is repeatable over longitudinal runs of data. And it's those individual differences in average telomere length which are predicting lifespan. So it's picking up some kind of early life environment or genetic effect which is associated with overall lifespan. And this would lead to the expectation that the associations between telomere length and survival should occur at the among individual level. So an individual's average telomere length across its lifetime will predict average survival and lifespan. And there is some evidence from some studies, particularly kind of wild mammals, that's consistent with this kind of selective disappearance idea, that individuals with shorter average telomere length are kind of disappearing from the population over time and with age. And these are really the kind of two models that we wanted to try and test in our sow sheep system. And to do that, we need a lot of longitudinal data, which is, I'll explain how we generated that shortly. But it's probably a good point to just stop and introduce you to this study system. These sheep are resident to a very remote archipelago. You can see it kind of pictured there. St Kilda is around sort of 50-60 kilometers west of the westernmost point in the Outer Hebrides off the west coast of Scotland. And it's composed of three main islands and a series of quite stunning sea stacks as well. And the sheep that we study are resident to this ringed area called Village Bay on the main island of Herta. So they live, as I said, in kind of completely unmanaged state. They're unpredated. And since 1985, the animals resident to this area on Herta, they've been caught and marked within usually a few days, but certainly within a couple of weeks of birth, so they can be individually recognized with unique ear tags. And then they're followed very closely throughout their natural lives, and all kinds of information is kind of gathered on them. So this study's been running for over 35 years now in a very consistent manner. And long-term individual-based studies like this, they involve huge input and kind of efforts and energy from all kinds of different people. It's impossible to sort of attempt any full meaningful acknowledgement of all the people that have contributed to this study. But I wanted to single out three individuals in particular who I think have been instrumental in the kind of success and the maintenance of the kind of quality of this incredible study system. And I should really stress that my role in the kind of fieldwork side of this has been very, very small. Josephine Pemberton, who I mentioned before, who's a colleague in Edinburgh, who's been managing and kind of running the project for the last, I think, 2 or 3 decades. Also, very important to mention Ian Stevenson. Ian's been involved in the project almost since its inception in the late 1980s. And Ian set up our kind of bespoke project database and continues to kind of manage and kind of update that database. And really it's his kind of work on the data management front that gives us kind of the real advantage, I think, in terms of being able to wield this really complex data that we collect, lots of different kinds of data, making it very, very consistent in the way that we can work with it, and really making working with this very complex longitudinal data set a real pleasure. The last person I wanted to single out here is Jill Pilkington, who's the long-term field researcher out in St Kilda. She's been leading on the fieldwork side for over 20 years now. She's the person, like Ali and Sean on Rum, who knows the individual sheep very well, who's out there for, I think, around 6 or 7 months of the year, most of the time, and who really makes the fieldwork tick and deals with the many and very challenging logistical issues of doing fieldwork in this remote location. Also supported by the National Trust for Scotland, who own the islands, Kinetic and Kilda cruisers who help us with transport, and many researchers and fieldwork volunteers. What the study has really generated is an incredible longitudinal life history data set. Last time I looked, we had nearly 8,000 individual sheep for whom we had birth and death records. And the work that Josephine's done over the years with kind of molecular paternity and parentage analysis has provided an incredible amount of accurate information on the population pedigree. This plot on the right is an old Surrey sheep population pedigree. You're not supposed to take away from that anything other than we know a lot about the links between parents and offspring in this system. We've got data on 16 generations of sheep now. And as I said, we have this very kind of accurate population pedigree allowing us to do quantitative genetic analyses. One of the kind of critical things about the sheep system that really made it interesting for me from a point of view of senescence and has really driven my increased focus on this system over others is the fact that every year in August, a group of field workers, usually about 12 or 15 of us, go out and set up these quite remarkable series of kind of net walls and corral traps. And we try our best to catch as many of the resident sheep as we can. And we usually get between 50 and 60% of them. And what this means is that at capture, we can obviously take various measurements, morphological kind of health measurements, but also take blood and fecal samples. And over the last decade or so, we've been doing more and more with those blood samples and the fecal samples, and we set up this kind of pop-up lab and this beautiful little cottage called the Feather Store in the corner of our study area. But essentially what's been going on really since the early 1990s by Josephine and Jill is that every blood sample is separated into plasma and buffy coat fractions, and those fractions are taken back to Edinburgh and stored in large freezer archives. It's the buffy coat that's of interest from the telomere point of view, because that is really the kind of leukocyte cell layer. And again, at last count, in the freezers in Edinburgh, we had nearly 7,000 available samples, both plasma and buffy coat, from over 3,000 individuals. So these are samples taken across the lifetimes of individuals. And the work on telomere dynamics that we've been doing has been funded by BBSRC and has involved a lot of different people doing different things, particularly in the lab over the sort of, I think, 5 or 6 years that we were focused on doing this. So Jen Fairley is hugely important in kind of setting all the methods up and kind of overseeing and making sure everything ran smoothly in the lab. But big contributions initially from Becky Lister-Kay and then Sarah Underwood in the lab, many other people contributing. Lee, again, offering huge amounts of insight into what we were doing. And also very important to acknowledge Hannah Froy, who worked as a postdoc on this project, who was really hugely important in kind of overseeing sort of data checking, quality control, and also the analyses that I'm going to show you. We selected around 4,000 samples, so this is a subset of the total available buffy coats, and really this was designed to try and get as many as we felt we could logistically and maximize the longitudinality of the samples. Jen and co in the lab set up these really high throughput lab pipelines using a liquid handling robot that we were able to get access to do both DNA extraction and measurement of telomere length by monoplex qPCR. And I think these guys worked really, really hard to establish what I think are quite rigorous quality control protocols. So they measured DNA yield, purity, and integrity. And they also measured qPCR efficiency and repeatability. And working with Hannah, they really established some thresholds for quality control. And if samples failed those, they were repeated. If the repeat failed, they were left aside. So we ended up with 3,641 samples, which were led through into the analyses. They also did replicate runs to try and establish the technical repeatability of telomere length measurements in this kind of method. The most conservative kind of analysis that we could think of performing gave us a repeatability of 82%. Don't know whether people think that is high or acceptable. It certainly to us felt like an acceptable repeatability compared to other published studies out there on qPCR and also some studies of TRF. So this seems to us to be an acceptable level of repeatability. So what I'm going to show you is some of the analyses that Hannah did and that we published recently based on this data set, which really build towards understanding this relationship between telomere length and survival. So first, Hannah looked at the relationship between telomere length and age. And here we find an association that's consistent with the pattern we see in humans. So the animals are first caught at around 4 months of age in their first August, and then every year subsequently, we see this rapid decline in telomere length over the first year or so, and then a still significant but much shallower decline thereafterwards. And there's very limited support for sex differences here in this analysis. So having controlled for this effect of age, Hannah then went on to try and decompose variance in telomere length into different important relevant components. So things like annual effects, maternal effects, but also the effect of the individual, which captures the repeatability, the consistency of telomere length over time, over its lifetime. And Hannah found repeatability of around, so it's 24%, around a quarter of the variance that we see is attributable to the individual's identity, which is an appreciable repeatability in the kind of ecological studies. The effects of things like year and mother were actually really small, which is maybe surprising. And what Hannah was able to do was to use the population pedigree to decompose that repeatability effect into a genetic and a non-genetic component. And what she found was that almost all of the variance among individuals was attributable to genetic effects. So the heritability came out as being almost exactly the same as the repeatability. So the heritability of telomere length in this data set is 23%, which again, it is appreciable. The kinds of traits that we look at in the sheet, things like body weight, skeletal measures, they tend to be a bit higher than that. But life history traits, they will often have heritabilities in that kind of range. So this is a significant and appreciable genetic effect on telomere length in the system. What Hannah went on to do subsequently, which is statistically a little bit more gnarly, was to set up multivariate mixed effects models in which she included telomere length, survival, and weight. So this is body mass measured at capture in August at the same time as we measured telomere length. Now we knew, or we expected there to be a relationship between telomere length and survival. Shorter telomeres predicting increased survival. And we know from previous studies that August body mass is a strong predictor of subsequent overwinter survival. And what Hannah did was use these models to decompose where that covariance with survival was coming from. Was it coming from the among individual level, the annual level, or the within individual level? And that's what these plots show. So if we look first at this leftmost set of correlations, this is the relationship between weight and survival, which we know is strong and positive, being broken down into, first of all, the among individual level in green. So this is individuals with high mean weight across their lifetime having longer lifespans. The year effect is reflecting the fact that years in which sheep have high average weight are also years when they have higher average survival. And the residual effect tells you about the deviations of an individual from those individual and annual averages and how that relates to telomere length. So if your weight drops relative to your own average or the annual average, your survival would also be expected to decline. So this is telling you something about the within individual effects. And you can see that for weight, as we expect, all three of those components are positively correlated. So what about telomere length and survival? Well, here what we found is that it's only significantly correlated at the among individual level. So it's really telling us that the signal is coming from individuals with longer average telomere lengths living longer, and it's not coming from these kind of within individual deviations. So this is consistent with selective disappearance of individuals with short average telomere length. And it is inconsistent with the idea, I think, that telomere length is a marker of biological aging. If it was a marker of biological aging, we'd expect sort of telomere attrition, so loss of telomere length over time, deviations from these averages to be predicting survival. And it's really not, I think, consistent with this exposome-like model of the telomere length survival relationship. Again, because we have this very accurate population pedigree, Hannah could go even further and decompose that individual effect into a genetic and a non-genetic, so-called permanent environment effect. And here again, for weight, you can see that all genetic and other components are positively correlated. For telomere length, what emerges, this is kind of borderline in some respects, but I think most of the signal at the among individual level is coming from the genetics. It's not surprising because we'd already found that most of the among individual variation is genetic, but it's suggesting there that there's a genetic basis to the correlation between telomere length and survival. We also interestingly found what looks like a negative genetic correlation between telomere length and weight, which we haven't explored very much. We're looking at this more at the moment. It might be consistent with some kind of trade-off between weight, growth, and telomere length. But what I think this is consistent with is the idea that there might be some genetically based variation in some aspect of physiological overall frailty or robustness, which is correlated or picked up by average telomere length. So it's kind of overall consistent with this sort of frailty or robustness model, and it's particularly kind of indicating that it might be genetic effects responsible for this. But it's very important to kind of caveat the sort of magnitude of these effects, these associations between telomere length and survival. So if you compare the telomere length, the RTL survival correlations with those with weight, you can see they're a lot lower. So it's important to keep that in mind, that these are effects of quite small magnitude compared to a measure that we know and is well established as an important predictor of fitness outcomes. So to summarize the results in So Is Sheep, I think we found support for this kind of frailty robustness idea, with the covariance between telomere length and survival being driven predominantly by among individual covariance. And over and above that, the evidence seems consistent with that coming from the effects of genes rather than consistent environmental differences or early life environment effects. This is obviously just one question that we could ask with this large longitudinal data set on telomere length. So we are delving deeper, many questions that we can address. This is largely being led by two very talented PhD students, Sanjana Ravindran, who's working with myself and Jacob Morad in Edinburgh, and Florentine Rameau, who's doing a PhD in Lyon with Jeff Lemaitre. I've been lucky enough to co-supervise and try and persuade him to do some work on the sheep. Sanjana's been looking at telomere length and reproductive investment in females, and also the association between telomere length and infection and immune responses, while Flo has been looking at the reproductive investment, telomere length, and growth effects in males. And sadly, I don't have time to tell you about that work, but hopefully you'll be able to read about it soon. Happy to answer questions about it. Before I finish, I wanted to end by returning to the question that I said at the beginning of the talk. I'm not sure I have completely, I'm going to be completely able to answer this question, but reflecting back on my experience in this kind of area, I think there are some very interesting things emerging about telomere length as a biomarker. It certainly looks to be associated with past environmental experience and maybe stress in a way that could provide important insight in various ecological and evolutionary studies. It does look in many labs' hands to be repeatable and heritable, and it does seem in many cases to predict survival. So these are all very interesting and potentially important properties for any biomarker within this kind of field. But I don't think there's evidence I consider particularly compelling that short telomeres or telomere attrition play a causal role in aging or survival. I can't see any evidence really that convinces me in either the ecological or human literature that telomere length is a good biomarker of aging. I think we really need to reflect on that and maybe reconsider how we're portraying this as a biomarker. As I've explained, comparison across studies can be very difficult, and making sense of the patterns that emerge from different studies and different systems because of these methodological differences. There are an overwhelming number of studies at the moment on birds, and I don't think enough on other kinds of taxa. I think in particular, ectothermic vertebrates are a very interesting area that we should focus more on. Some of the results coming out from studies of fish and reptiles actually suggest that some of the relationships that we see in birds and mammals might flip. And we know that those species can express a lot of telomerase in somatic cells compared to birds and mammals. There's a lot more to be learned. I think one of the things that bothers me is that it's not entirely clear what aspects of physiological function or somatic integrity telomere length measured in various ways actually reflects. This is a big question, I think. And also, I think we're still a long way from understanding why telomere length varies so much among individuals and species. But a lot of very interesting work is being done. And I think it is a very, very exciting area. And hopefully will continue to be so. And with that, I'd like to finish and thank you all for your attention and happy to take questions. I'll try and stop sharing.
Thank you, Dan, for that wonderful talk. If anyone has any questions, feel free to ask them. Yeah, I see clapping virtually, so hear it virtually. If anybody is shy to ask questions, just write them in the chat and I will read them, or Dan can read them.
And I have a question. So in birds, there was an early result by Mark Hausman and group that longer living birds seem to be protecting their telomeres. So telomere loss was less per annum for, like a storm petrel than for a zebra finch. Do you find that in mammals too? And especially in if you compare it to humans, which age very differently from all mammals?
I mean, I don't think we know. I don't think there's enough data on mammals yet. I think the work that Mark's done is really important because he's studied a lot of different bird species using a very consistent and very robust methodology in the lab. And I think so his work really provides some of the most compelling evidence of that sort. I'm not aware, I mean, others here, including Britt, might know more than me, but I'm not aware of sort of the similar kind of setup in mammals that will let you address that kind of question. So I don't know.
Sure, thank you.
Then, do you have any regarding the biphasic reduction of telomere length? So why does it happen?
My understanding is that it's to do with, I mean, obviously it's kind of a rapid growth phase, which is obviously kind of associated with kind of driving telomere cells, lots of replication, and that may be responsible for it in part. I mean, it's interesting that that's a kind of that is something kind of a sort of dogmatic thought on how telomere length should change with age, and it's what we see in the ruminant systems that I've worked on. That's what you see. So when you have enough resolution in early life, you can see a more rapid decline. But Florentin, who I mentioned at the end there, the PhD student working with in Lyon, he's done a meta-analysis which I think was published very, very recently, looking across non-human vertebrates, and actually there isn't support for that at the moment. So you actually can't find a particularly clear difference in the rate of decline in telomere length between early and late life. So I'm not sure whether that actually generalizes or whether it's more to do with problems with the quality of data across the different studies. But I think there's a kind of question about how and why age-related declines in telomere length vary. I don't think we necessarily know. But the human studies are pretty clear, and I think some of the larger longitudinal mammalian studies also find that pattern.
So it cannot be attributed to like a juvenile survivorship bias or something. Animals.
It could be. So we've certainly seen in the sheep there's some suggestion that selective disappearance is contributing to some of the change. But when you look at longitudinal data, you can see it very clearly that it is longitudinal. There's an actual decline. I don't know the human literature looking at early life longitudinal dynamics, whether they've demonstrated that to be the case as well. But we can definitely see it longitudinally in the early part of life in sheep. And we have looked at, we get samples, they're very small blood samples. We get samples from the neonates when they're caught around birth. And again, if you put those together, you see an even sharper decline. So those neonate telomere lengths are even higher.
Interesting.
Thank you, Dan, for the very interesting talk. I would have a question. We briefly talked about it before the talk, actually, but I would like to ask this: as you, as an expert of multi-level variance, here we're dealing with multi-level variance, because when you're measuring telomere length, you're measuring it from a population of cells from the blood, and especially in birds, where you also have leukocytes and you have erythrocytes, they all might contribute to what we're measuring. And so I would like to ask, how can you deal with this kind of possible intra-individual source of variation in telomere length?
I don't know. It's incredibly difficult. It really does. In the literature, it gets swept aside a lot of the time, and it's a really potentially very big problem. So we've often worried about whether what we see could be driven by differences in the leukocyte, the cell pool. So that it's well known in studies, sort of cellular studies and human studies, that different kinds of leukocytes have very different telomere dynamics. We've done things in the past, like doing really kind of crude cell counts and trying to relate them to telomere length. It's not very satisfying. Basically, you need to do cell sorting, I think. So there's flow fish techniques can actually do this, but we've never been able to get anywhere near that kind of approach. I mean, I don't know whether others have used it. So you can look at individual cell types. I have talked with various people about the possibility of trying to separate in some way the red blood cells and white blood cells. I think Matt Olsen has done something like this in reptiles, but I think it was also very difficult to do, and it's not really been done very much. So the answer is we don't know, but it's a very, very important thing to try and tackle. I mean, it could be really important in understanding the differences that we see across systems, I think.
Okay.
That's not a very satisfying answer, I'm sorry.
Thank you. We have a question from Michaela. How?
Hi, Ella. You there?
Sorry. Hi, Dan.
Hello.
Fantastic talk and fantastic study. This is amazing.
Thank you. It's nice to see you. It's been a very long time.
Yes, it's great. I wondered, I mean, you first showed us the difference, I mean, some of the differences that you get when you use the TRF assay as compared to the qPCR assay in precision. And then in the study on the soy sheep, I mean, very understandably, you used qPCR and it was probably, you know, still an amazing effort. But can you comment on the possibility that you reject one of your hypotheses, you know, that you weren't able to actually get a signal of within individual differences because you used qPCR?
It's entirely possible. So it's this like inevitable trade-off between, I mean, there is no way we could have generated anything like that sample size with TRF. So there are labs that really do seem to be able to put out big TRF data sets, but I don't think they're anywhere near as big as the one we've used. And so you're kind of left in a situation where do you invest in using TRF and accept you're going to have smaller sample sizes, or go with qPCR, and it means you can go bigger. I don't know. I mean, there are people doing power analyses of various kinds addressing these kind of questions. I don't know, Daniel Nettles in Newcastle seems to be publishing quite a lot on this at the moment, and some of the papers he's publishing are really quite scary in terms of what happens with qPCR. I don't know, it is tricky. So you can't be that confident that there aren't within individual associations there. But I think what it probably tells us is that the among individual in our data set are bigger.
Okay, cool. Thank you.
I would be reasonably confident in that. And I actually don't think, from what I understand about the kinds of models that Hannah ran, I don't think we could have generated a TRF data set that would have coped with that model. So you
Sure, I just wondered whether you did some additional statistics, you know, throwing in, I mean, I know these models are complex by themselves, but, you know, throwing in some uncertainty with the method or something, because I also noticed that, you know, the row and the plate also made a big difference.
Yeah. So we did put those in the models. So the kind of everything we could get of a sort of technical nature that might be things, we put it in the models, which is a really important thing to do. And they are making a contribution. So we found even with the robot that you get plate effects and you get position effects as well. This is well known with qPCR. I think it goes on in TRF as well, but people, it's not so much discussed, I think. So I mean, you're kind of doing the best we can. And I do think there's always the issue with power and qPCR because of the way it's measured. It compounds error in a really profound way. I think what I would take from it is if you want to do qPCR, you have to go big. And one of the problems we're seeing with the meta-analysis is small qPCR studies. You're probably going to find, like, who knows what you're going to find, basically. It could be anything. But you see really beautiful TRF studies, for example, that are quite small in sample size that look like they've nailed something. So I think that what I've learned is that you really have to try and get your head around the pros and cons and make decisions based on the kind of question you're trying to ask. That both methods have a very important place.
It's still a fantastic study. Yeah, thank you.
Thank you. Steve Smith is up, afterwards Britt Heidinger.
Hi, Steve.
Hi, Dan. It's nice to see you again, and thanks once again for a wonderful talk. I remain constantly jealous of the study system that you have there with the soya sheep, and it's great to see more and more interesting data coming out of this study. I just wanted to get your comments on using telomere dynamics rather than point estimates of telomere length itself. I mean, you've concentrated a lot on the genetic differences in telomere length between individuals, but do you think we can get more information when we use changes in telomere length over time per individuals in terms of looking at questions about biological stress and survivability?
It's difficult. So the kinds of analyzing change on that kind of scale and that kind of data set, we've mucked around with different things, and it tends to result in like some quite, again, you've got error compounding in the way that you actually just estimate telomere by qPCR, and you have a similar problem when you potentially when you look at change. For me, I think that kind of method that Hannah has really kind of pushed and kind of championed with both this and other kinds of physiological data we're using for large longitudinal data, where you don't necessarily, it's not experimental, it's observational. So you're kind of asking, well, what kinds of things could be influencing the dynamics here? I think it's and the associations. I think it's really powerful. I think change, if you have an experimental input or something quite explicit about the environment or something like that you're interested in looking at the impacts on telomere dynamics, then that's really where that could come into play. I think there are limitations in what we're doing in terms of what it tells us about those short-term dynamics. I know, like, some of your work really interestingly kind of speaks to the kind of question of telomere elongation and what's going on there. It's not something we can speak to really with the kind of analysis we're doing. So maybe it's a bit crude and kind of broad, but I think we were sort of struggling with the sheep initially to kind of frame, like, we expect this environment, this sort of environmental variable to impact telomere length in this kind of way. So we kind of step back from that.
Yeah, I think you're right. I mean, a lot of it comes down to the question you're asking, I guess, and to see what information you can gain at rates of telomere loss can be quite powerful, I think. Yeah.
It's something we could look at, particularly it could be interesting in early life. I think that's what a lot of the quite exciting bird studies have done recently, is they kind of looked at the rate of loss in the kind of nesting phase and found some quite interesting associations with that. So again, something to come back to and look at. It's a really good idea.
Yeah, and combining it, I guess, with studies of telomerase and looking at telomerase activity is important as well, because it does seem to be the sort of trade-off there in costs for being able to activate telomerase as a sort of maintenance phase.
Yeah, I mean, I think the one thing about working in mammals is you're looking at immune cells. So again, this is why we're trying to relate telomere length back to kind of what we know about infection and immunity, because it should be related, and that again could be quite interesting. I don't know, it's not throwing up particularly strong relationships or in the expected direction, to be honest, but I think that's quite an important thing to think about when you're looking at the white blood cells specifically.
Okay, thanks for that.
Thank you, Britt Heidinger. The floor is yours.
Okay, thank you very much. That was really a fun talk to listen to, Dan. Thank you very much. So I have sort of a two-part question. The first thing I wanted to ask you, kind of on a very different level, is how would you define a biomarker? So what would, if you were going to say what a biomarker was, how would you define it?
That's a good question. I don't think I've got an answer for that, but I think I'm thinking about it more from the biomarker of aging type perspective. That what I'm thinking about is some measure, some marker that is reflecting in some broad way what's happening physiologically in later life in terms of deterioration or maintenance of function over and above chronological age, which is usually how they define biomarkers of aging. Generally, for me, biomarkers, I guess what I'm driving at is you're measuring something. Usually, you're talking about something of a physiological nature, which is kind of probably indirectly related to some things that you're very interested in. So a lot of the immune measures we take, the more we kind of delve into them, you realize they're biomarkers of what's going on immunologically. They're not actually getting in and directly measuring how the immune system is directly kind of causally influencing things, but they're kind of reflecting important things about the way the immune system's working. I don't know if that's the satisfactory answer.
Well, it's more that I'm trying to understand the distinction between the two hypotheses. So the one that you presented, the frailty hypothesis, it's that there's some genetic difference among these individuals that influences what their telomere length is like, and also influences what their survival is like. Or it could be that these individuals experience conditions throughout life that change telomere length over time, and that is related to survival, this exome. And that's the hypothesis you don't find support for, right? You find support for the frailty hypothesis. And I guess I was interested, and I was wondering, sort of depending on when the animals are sampled, those two hypotheses wouldn't have to be mutually exclusive, right?
No,
you may have found evidence for both.
Yeah.
And I also wondered if in the sheep, could there be something that could be happening early in life where I think you're acknowledging that, you're putting it in with the frailty hypothesis, that there are these sort of persistent effects that play out long term, but it could be that they're experiencing some stresses early in life that are being reflected in this telomere length that we're calling frailty, that they persist, that it's, it could be genetic, it could be environmental early in life, but it's persistent.
Yeah. So we first measured them at 4 months. So there could be all kinds of effects during gestation or that early period of life that could shape the telomere dynamics that leave some kind of consistent mark on. Yeah. So certainly wasn't meaning to suggest that those are mutually exclusive. You might expect them both to be there. I mean, in fact, some of the things that we've done been quite surprising how much they differ depending on what you look at.
But would you not call it a biomarker if it was those things that happened during early life?
Yeah, I would. I think it's
you would.
Still,
okay.
Yeah. It's kind of reflecting something. It's all very nebulous, isn't it? But it's kind of reflecting something that is of interest. We don't necessarily. The problem with telomere length is we don't necessarily know what it's reflecting. What is it physiologically about physiological function or that it's telling us? Probably varies a lot depending on the system you're studying, whether it's white blood cells or red blood cells. It makes it quite a difficult biomarker for me to get my head around, but there's no doubt it does correlate with some very interesting things.
But if you had instead in the analysis found that there was this large environmental effect on telomeres rather than a large heritable component, and that environmental component had been related to survival, then you would have landed more on the end where you felt like it was maybe a important biomarker, or it was telling you something about the exome. Is that true?
I suppose from the point of view of something about telomere attrition reflecting past environmental experiences, like across life in some kind of cumulative way. I think for that to be supported, you need that. You can have both covariances, but I think you need that with individual covariance for that to make sense. Yeah.
Okay, great. Thank you very much.
Thank you. We have a question in the chat. Sawadi Francisco Nduguru, sorry for mispronouncing the name. Why so many studies on telomere length and senescence or aging in birds than other taxa? what is so interesting with birds that attract researchers?
I don't know. I'm probably the wrong person to ask that question to. I don't. I think it's true in a lot of these kind of, most of the like high quality long-term individual based studies are in the wild are done on birds. Just the I think it's in some respects easier to study them in that way, and I think a lot of kind of interesting ecophysiology and this kind of work is going on in birds. Yeah, I don't know. Britt might have other things to say about that. You do see kind of when you look at kind of the evidence for senescence, you can see it's quite balanced between mammals and birds, but actually the more physiological work that goes on, I think it tends to be more focused in birds. I'm not entirely sure I know why that is, but maybe it's to do with the ease of sampling. It's definitely something that many people in the recent literature are saying is it's important to have lots of information. Some very cool comparative work being done on birds and very exciting papers coming out, but we're kind of missing something by not having better representation from other groups.
I'm fairly sure that it's something to do with catching the birds. I mean, it's pretty easy to do it.
Yeah, not so easy to catch sheep, I tell you that. Well, I don't know how easy it is to catch birds, but the sheep can be a bit of a problem.
Yeah, probably this could be a problem for longitudinal data in birds and wild birds at least, which is more easy to do in mammals. So we have another question in the chat from Csongor Vágási from Cluj.
I can read that. Is there compelling evidence that oxidative stress increases telomere attrition either within individual or among individual? Or maybe, no. There's some. I'm not sure there's particularly compelling evidence. I don't feel like I was saying this to Adam and Attila before that we kind of finished the work in the lab on this a few years ago now, and in preparing the seminar, kind of returning to the work and the telomere literature, it's really amazing how many papers are coming out in kind of non-model systems and kind of ecological systems and telomere length. It's impossible to keep up with the literature for me anyway. So I don't feel like I would be well placed to answer that question. But my feeling is there are some studies that suggest there might be that kind of relationship, others not. And I don't know at the moment whether there is really compelling evidence that there's a clear relationship between oxidative stress and telomere attrition.
Thank you. Are there any more questions? Okay, if not, Pablo has a question. Pablo from Pat Monaghan's group.
Hello, how are you?
Hi, Dan. Fine. Nice talk. I have a question a bit following Britt's on considering telomere length a biomarker or not. And do you think it could be because of how difficult it is to establish the causality, I mean, to manipulate telomere length somehow? Do you think that will be the reason behind that? Even longitudinal studies, I mean, they're not gonna ever tell us of causality at the end. They can get something close by, but do you have any input
totally
on that?
I mean, you can't infer causality from these kinds of studies. It's I think generally very difficult. So there are papers coming out. There's been a few papers from Mary Blasco's lab recently where they are obviously in rodents trying to manipulate telomere dynamics very hard to try and establish whether they're causal effects. I don't know. I mean, they have some evidence to support that. I'm just not. It's so far removed from the kind of context that I'm used to thinking about and working in. I find it hard to put those kinds of studies together with the sorts of processes that we're dealing with. Maybe it's just impossible to establish causal relationships. I find it easier to think about it as reflecting something important that's going on physiologically. I think it would be really cool to sit down and talk and think about how we could get closer to understanding what it is and whether it varied. So, for example, is it something different in birds and other vertebrates to mammals because of the kinds of cells we're measuring it?
Actually, I was wondering if you have thought about that. More is a redundant reflection. I mean, what we see in telomeres for sure on the red blood cells is just a redundancy of the cell turnover. Mm-hmm. And perhaps it's not telling us about the functionality per se, perhaps, because it's just mostly doing growth. I mean, of course, could be that, or.
Yeah, I mean, again, it's possible. Again, I really worry when you're looking at leukocytes. It's something about the the way the white blood cells are behaving and what they're having to do. So certainly some of those cells can express quite a lot of telomerase. They can reset their telomeres when they go into a kind of activation and proliferation stage. So I think it's very, very hard with these kinds of studies where you're measuring at such a crude level to know what's actually going on. And again, it would be lovely. We did have discussions and play around with using these much more kind of refined methods. So we talked to Duncan Baird about Stella. Jen did have Stella working in the sheep at one point, but we never followed up. It's so hard to do, and it's so hard to interpret because you get like the individual chromosome telomere lengths. You've got a distribution of telomeres like that, and the kind of flow-based techniques where you could separate the cell types. It would be amazing, but I just don't know that we could do it.
Thanks. Yeah.
Do you know something about the telomerase activity? If you can measure it somehow?
You can measure it. There are assays. We've never used them, but there are people looking at manipulating telomerase and measuring it as well in different kinds of settings. So it is possible to look at that.
Yeah, there's a few methods coming out nowadays. There's one we're working on in my lab that uses real-time PCR to look at the activity itself of the telomerase. So you actually take the cells themselves and then you lyse them, and then you add to that the basic components for telomere elongation, and then you measure that elongation itself to see whether there's activity occurring in those cells.
Cool. Well, that sounds really neat.
Thank you. Second, the class is starting in the room where I was holding the seminar.
There's a question from Pablo asking, in your study system, do you know if there are differences between white blood cell types? Because yes, there are certainly. We've seen changes in the proportion of different kinds of white blood cell types over the lifetime of the individuals, and you'd expect there to be the immune system and its kind of makeup does change dramatically with age. We have done a few things where we've tried things like kind of counting different cell types with hematology, kind of microscopy, and correlating that with telomere length, and we didn't find any relationship with kind of overall leukocyte telomere length. And I mean but that's really crude, but it may be that it's not being driven by one particular cell type. But I think yes, the simple answer is yes, and we don't know how that plays into the very crude leukocyte telomere length measure that we've got.
Up because a seminar is starting, another seminar in the room where I was.
Questions or comments?
See the question in the chat room.
Okay. There's a question. Dan?
Ooh.
I don't know what's going on until I was recording or not recording.
It's okay. You can stop.